Risk, Validated Methods for Cleaning Reusable Medical Devices, brought to you by Ball Seal Engineering and Design World magazine. My name is Paul Heaney. I'm the editorial director for Design World. A little background on me. I'm a mechanical engineer from Georgia Tech. I've been covering the engineering and manufacturing worlds for more than 15 years, and I'm pleased to be your moderator today. Now, just a couple of housekeeping details before we get started. If you wish to tweet about this webinar anytime during or after, or join in on the conversation, you can use the hashtag DWWebinar, which is all one word. We will have a Q&A session after the presentations, so please go ahead and submit your questions as you think of them, and then we're going to ask as many of them as we can after the presentations are finished. Questions can be asked using the GoToWebinar dialog box right there on your screen. I'd like to take this opportunity to thank our two presenters for being here today, and I'd like to introduce them now. Stephen Twork is the Global Market Manager for Medical Devices at Ball Seal Engineering. Steve has been working in the mechanical sealing and connecting industry for more than 30 years. His specific area of expertise includes orthopedic instruments, surgical power tools, and implants that utilize the company's seal and spring component technology. And Emily Mitzel is a laboratory manager at Nelson Laboratories, a microbiological testing company. Emily's expertise includes reusable device reprocessing validations, and she is presented at AAMI FDA, as well as seminars, trade shows, and medical device facilities throughout the world. Emily is an active committee member of many working groups with AAMI and ASTM. She holds a Bachelor of Science and a Master of Science in microbiology. And now, without further ado, I'm going to hand the mic over to Steve. Steve?
Thank you, Paul, and everyone who is participating on this webinar. I will briefly review why Ball Seal is participating in reducing risk for reusable medical devices and steps we have taken to develop baseline data on common sizes we apply today throughout the medical device industry. Plus, our plans on adding more baseline test data using new cleaning methods and alternative hardware solutions that may support more challenging geometries for mechanical connections. And then I'll hand it off to Emily, who will provide a bulk of the presentation with valuable insight to meeting standards and guidelines for validating reusable medical devices. And finally, I will summarize with a few points followed by Q&A, and we should get this all within 60 minutes. I also want to point out this is Ball Seal's first of many webinars on sharing relative information on important industry issues. So we're excited about this communicating in this forum. So my first slide is a list of 3 key points of awareness, which are eliminating these nasty infections by sharing relative information on manufacturing levels. As the FDA has stated, this is everyone's problem, and Ball Seal is investing in baseline testing to increase our awareness by keeping up with cleaning standards and methods for challenging simple connections. Our chemical springs are used and applied to over 500 types of medical devices, so there's existing validated methods in practice today. So here's an image of our actual test samples used in our baseline tests completed earlier this year. We used our most common springs and bore sizes in stainless steel housing materials and designed for a worst case by using a housing-mounted spring inserted into a single housing with multiple groove types, like the one you see here in this image. The only access points to flushing is from the ID bore. We also use no flush ports or special designs that may assist in improving cleaning results. Again, we only used common springs and sizes and designs used today. Then we took our samples to Nelson to establish baseline testing in accordance with the Reusable Device Guidance AMI-TRR 30, which Emily will speak to in more detail later on this presentation. And we chose more of a low-level cleaning method, which is a manual operation by simply soaking, scrubbing, and rinsing, then left to dry. And as a result, we showed no visible soils under normal lighting conditions, and the hemoglobin test came well within acceptable levels. There we go. Also, we also did a hemoglobin as well as a protein test. I'm sorry here, little difficulties. We did a hemoglobin test as well as a protein test to ensure that we're meeting cleanable requirements. And if you need more information on detail on this report, please contact your friendly microbiologist. And of course, I will email everyone later a copy of this final report, as well as the detailed procedures that we use for referencing. Our next step is to test more 5mm housing bores in the same configuration using automated washer disinfecting methods, as our first method was just purely manual, which is pretty common today. So we'll contact everyone present in this webinar via email on all future updates related to this presentation. Let me try that. Sorry, there we go. So we talked about alternative solutions a little bit, where you can enhance the cleaning by using OD and ID flush ports, as well as open grooves and product alternatives. So we can be more creative with our groove geometries to enhance flushing if you choose to use ports and trough grooves. So let us know if you can, if you plan on doing this, we'll help you with some feasible designs. You can see in the top right here, we have a piston-mounted or housing-mounted groove with some ports in there, as well as a piston-mounted spring groove. And that pretty much sums up my brief review of what we've done in testing and baseline testing with Nelson. And now I'll pass this over to Emily.
Well, good morning or good afternoon, depending on where you are located in the United States. I'm excited to be a part of this topic. It's a hot topic with all medical device manufacturers and the FDA right now. And Ball Seal is really on top of things. Really, it's a complex, really working with complex devices and making sure that they're easily reprocessable. And that's very important these days. All right. I've got a couple photos here, and there are some problems with cleaning reusable medical device. This photograph shows human tissue that was found in an arthroscopic cannula during an infection outbreak investigation from the University of Chicago Press. This next picture shows not necessarily a problem with the cleaning process, but a bristle that was left after the cleaning process. So this device probably is perfectly clean, but part of the cleaning process got left into the device. And this next picture shows probably what we would consider a rust inside the device in a surgical suction tube. And here you can see a internal part of a surgical suction device, and this is a picture from the University of Michigan Health System. And these devices were cut apart, and many of them were found to have debris inside the devices. Just a couple years ago, there was an NBC special on cleaning, disinfection, and sterilization of reusable devices in the healthcare facility. And this really showed an issue with patient infection control and infection rates that are on the rise due to more complex devices in healthcare facilities at this point. I'm not going to show this video today, as we have a shorter time frame today, but you definitely can go look that up and see that video. So the topics today that we're going to be talking about is a lot of guidance for reusable medical device manufacturers, the information that came out of the roundtable discussion, How Clean is Clean, not quite a year ago, some information regarding IFUs, device design for human factors, and we're going to be going into a full cleaning validation considerations. Some important guidance that has just recently come out in the past couple of years is the draft guidance for industry and FDA staff. This is a very interesting information from the FDA. This is still in draft guidance. They are still working on getting the more formal final approval for this guidance, but they are following this guidance, every part of the guidance, and so we have to make sure that we are complying with all parts of that when we submit information to the FDA. The AAMI TR12 and TR30 also have to do with reusable medical device reprocessing and have a lot of good information regarding some base mark levels and so on. So that is very important information to follow as well. Currently, there are many AAMI groups related to reprocessing. This is, like I said, it's a very hot topic, and there's a lot going on with all these groups. We are working on revising standards. There are standards that are finalized. We are working with creating new standards and so on. And so there's a lot going on with the world of reprocessing at this point. There's also a lot of new guidance coming out. The human factors device reprocessing is in a draft. The standard instructions for use is also in a draft format. We are also defining quality systems for device reprocessing for healthcare facilities, and this should be in draft format by the time we have our AAMI meetings in October. There's the endoscope reprocessing document is getting to be in final form at this point, and we're also working on low and intermediate level disinfectants and their use documents. There are also new working groups from ASTM, and we are just finalizing the new guide for standard test soils and validation of cleaning methods for reusable medical devices. That is going to be a great guide for how to determine what kind of test soil should be used during cleaning validations. Last September, we had a How Clean is Clean roundtable discussion. I was able to facilitate this discussion. It was very interesting. The participants included AAMI staff, FDA, manufacturers, test labs, and hospital staff. And we were really trying to determine really how clean is clean and what is the maximum amount of residual soil that can remain on a medical device after cleaning and still render it safe for patient use. So really, there's a lot of parts that go into the cleanability of devices. And some of these are device design, device compatibility with the systems, the washing systems, detergents, disinfectants, sterilization processes, etc. Also, the instructions for use, how easy they are to follow and so on, and that leads into human factors on being able to follow these procedures in the healthcare facility central processing. So really there's two ways that we are defining cleaning. Manufacturers must first validate the cleaning method that will render the device safe for patient use. And then healthcare facilities need to find a way to verify that the device has been cleaned after each patient use. A few other things that should be noted before we get into the main validation of cleaning is that one test soil cannot be used for all medical devices. And we'll talk about clinical relevancy and so on later in the presentation. Manufacturers should conduct validation testing on devices that are in a used state. So these should not be brand new devices, so that they don't, so that they have a few cracks and crevices and so on. Manufacturers should ensure that the healthcare facility can correctly clean the device, and that the instructions should take into account limits of the healthcare facility and also limits of the device itself. So some of the instructions that we use to make our instructions for use should be that all the instructions are, in the instructions for use, are grammatically correct. Everything should be very legible. It should be presented in a logical order. In the first step through the last step of the reprocessing steps. Everything should be in a short, concise voice, and also it should show images to show essential steps. The use of language such as a minimum of, if appropriate, if possible, and so on has been very discouraged and is no longer acceptable. It not only allows for misinterpretation, but steps may be missed during the process. Disassembly is necessary for a lot of devices at this point because of the complex devices that we are seeing in healthcare facilities and used in surgical procedures. Disassembly and assembly steps should be outlined and included in the IFU. And this should include illustrations, diagrams, descriptions, and videos to help any part of the system to help the healthcare facility perform the right steps on the devices prior to the cleaning steps and after the cleaning steps before the sterilization process. Other very specific instructions that need to be included are the detergent dosage, temperature, the water quality, the type of cleaning instruction, the brush type and size, and even a part number, and then every type of specialized supplies that are utilized in the cleaning procedure. So there's been a lot of new FDA expectations for manufacturers over the past year and even more so, but this is really very specific on human factors, and this has become more pronounced in our clients that we've been working with over the past year and so on. One of this is that manufacturers are encouraged to provide on-site training and education for central processing staff. And then this also includes open communication and encourages collaboration on device design to make sure that we are meeting all the functionality requirements and also addressing human factor considerations. We also, in this part of the process, we need to consider feedback from healthcare facility personnel, sales rep, etc. We need to solicitate input from individuals knowledgeable in the processing equipment and methods to ensure complete and accurate analysis. We also need to develop education, training, and competency verification materials for healthcare facilities. I know a lot of healthcare facilities have these processes in place, but medical device manufacturers are the ones that know their devices the best and can help with this even more so than even healthcare facilities putting this information together. And when we talk about complex devices, we need to really keep in mind the end processes and what needs to happen in between patient use. And so that includes provisions for cleaning and sterilization. And we need to consider this in the device design process to make sure that every device is cleanable and sterilizable. I just want to go into a few design features that make cleaning difficult. These design features may harbor unwanted organisms or organic material, and if we can't get all this organic material out during the cleaning process, it may lead to a device that is not able to be sterilized properly. A few of these design features are braided or twisted wires, textured surfaces, hinges, springs, dead end lumens and channels, inaccessible cracks and crevices, mated surfaces, sharp internal corners and angles, lumens, and O-rings. And there's a few design features that may have an effect on thermal conductance. These include aluminum-based materials and pliable materials such as silicone and rubber. Some materials may not heat up and have as good sterilization capabilities as others. So this needs to be kept in mind when we determine what materials we use on medical devices. Another thing to think about is all the number of components that make cleaning difficult. If we are disassembling devices and there are a lot of pieces, there is a greater opportunity for errors. Devices may not be reassembled properly, parts could be misplaced or intermixed, and there could be confusion which extends processing time. And from what I know and hear from healthcare facility workers is that they reprocess, thousands, if not, hundreds of thousands of devices every day. And so we need to make sure that all of their devices that they get through the system are quickly and as quickly and efficiently as possible. So as we talked about, complex devices really really make an unavoidable problematic design features, which may require more stringent cleaning procedures, such as mechanical or automated cleaning methods, and it may also require increased sterilization cycle times and drying times. So now I'll get into how to perform a cleaning validation. Some things we need to think about are worst case testing, appropriate test soil, and appropriate residual testing. So now the big hot topic for FDA is to make sure that you are using relevant clinical test soils. And we really need to ask ourselves what test soil should be used in a cleaning validation. And to determine this, we need to determine, will the device be in contact with blood, mucus, cerebrospinal fluid, brain tissue, etc. We also need to make sure that these test soils have the appropriate contents, viscosity, and tenacity of the above materials. And then manufacturers must justify why the specific soil was used and make sure the test soil is appropriate for all markers to be measured. We next need to determine the contamination method. This is determined by how the device is utilized in the clinical or in the surgical setting. Is the device immersed fully? Is the device handled by the physician's gloves, etc.? Then we need to simulate the use, so we need to actuate the device, how it is used in the surgery. And then we need to determine how long the surgery is. So how long should the device be actuated and set into the soil? And then what is the wait time between the surgical procedure and the cleaning process? How long does it take for the device to get from the surgical setting into the central processing area, or how long will it be cleaned after that point? And we need to make sure that we're applying this to the cleaning validation. So there are a lot of different methods of cleaning. All the procedures performed are exactly per manufacturer's instructions for use. We need to make sure that we are following the exact cleaning procedure when we validate the instruments so that we can make sure that it is followed exactly at the healthcare facility. There are mainly two methods of cleaning, and that would include a manual cleaning method, which includes soaking, brushing, flushing of the devices, and then an automated cycle, which is also mandatory if the device can handle an automated cycle, and that includes the washer disinfector. The cleaning markers that can be tested for a cleaning validation include protein, hemoglobin, total organic carbon, carbohydrates, detergent residuals, endotoxin, and bioburden testing, or bioload reduction. The three most commonly tested and most accepted markers by the FDA include the protein, hemoglobin, and total organic carbon right now. When we talk about acceptance criteria for a cleaning validation, we have to specify what we want for the acceptance criteria prior to performing the cleaning validation, and we must explain why we think that this is appropriate. AAMI TR 30 has a good benchmark criteria. This was developed for endoscopes many years ago, but it is really followed by the majority of medical device manufacturers and by the FDA at this point. A lot of the information that we need for cleaning validations includes all the description of the accessories required, every single technique that's used in the cleaning process, which includes all of the brushing, flushing, and rinsing. We need to know the water quality used for each process, and also the concentration and type of each chemical and detergent, and the exposure time and temperature of each step. So I just want to go over 7 criteria for reprocessing instructions, and these come straight out of the FDA guidance document, and I just want to specify these because they're very important. First of all, the labeling must reflect the intended use. There must be a thorough cleaning process for each medical device. It must contain a microbiocidal process. The reprocessing steps must be technically feasible. We must use legally marketed detergents and disinfectants from the FDA website. The instructions must be comprehensive and include reuse life testing and the instructions must be understandable by healthcare facility central processing personnel. Thank you, and I will hand the time back to Stephen to finalize everything that we talked about today.
Well, great. Thank you so much, Emily. That was very informative. Just a quick summary so we can get into the Q&A side of it. Improved awareness on cleanability, that's what this is about. And better understanding of cleaning methods and expectations, I think we hit some of that. And as well as, our chemical springs is, what we showed earlier was just one solution to meet a standard guideline. And of course, there are alternative hardware configurations for further enhancements that Ball Seal can do for groove configurations. And now we can get into the Q&A portion. So hopefully we have some questions that came up during the webinar that we could address.
Yes, sorry, I was trying to get unmuted there.
Okay.
Thank you so much, Steve and Emily.
Sure.
We're going to use the remaining time for questions. We have some that have been coming in, but it's not too late to send in your question. Simply use the dialog box on your GoToWebinar screen. All right, let's see here. The first one, this one's for Steve. Have you tested cleanability of piston-mounted springs? If yes, what were the results? And if no, to what extent would you expect the results to differ?
The answer to the first question is no. We have not tested piston-mounted springs yet. If no, what do I expect to differ? I'm not sure. I think it depends, on the cleaning methods that are used. So we would have to do some testing to find out what that really means, whether it's a manual clean or an automated clean. So I wouldn't be able to answer that one directly.
Okay. Emily, I believe this one is oriented to you. What residuals would you recommend for testing for a device that would be used for a hip replacement?
Yes. Let's see. For hip replacement, we would want to make sure that we are using a blood soil that would contain all the components that the devices would come into contact during that procedure. And we would want to make sure that we fully immersed or handled all the devices and actuated them appropriately, exactly how they would take place in the surgical procedure. We would work with that medical device manufacturer and have them explain to us fully the surgical procedure so that we can mimic that appropriately.
Okay, great. Another one for Steve. This says, we autoclave all instruments. Do the springs need to be cleaned via the recommended procedure in advance of sterilization?
I'm definitely not a microbiologist, but I would assume yes. I would hope that all instruments were cleaned prior to sterilization. Emily might know more about that than I would in terms of instrumentation cleaning processes.
Emily, do you want to add anything?
Yeah, Paul, could you repeat that, please?
I said, we autoclave all instruments. Do the springs need to be cleaned via the recommended procedure in advance of sterilization?
Yes, everything needs to be cleaned in advance of sterilization. We want to make sure that we remove all clinical soils or any environmental components off of devices prior to the sterilization process.
Okay.
Thank you.
Kind of a related question here. How susceptible is the spring to damage during cleaning, and what approaches do you recommend to minimize damage to the spring during the cleaning process?
Well, again, I think it goes back to the method used to clean. A manual method with using the wrong brushes or untrained techs could result in damaged springs during cleaning. So verifying and validating your cleaning methods is probably the best approach. I couldn't tell you whether we've experienced that or not because I don't clean devices. I'd have to go to the experts for that. We're just trying to understand it all, and maybe automated dishwasher would seem less likely to have damaged approach for that method. So again, I think it depends on how you validate and verify your cleaning methods.
All right. Emily, what test soil should be used for a device that's used for eye surgery?
So what we would look for that is having them, again, explain the surgical procedure to us exactly. And from the testing that we've done previously on instruments that are similar to that, the majority of the components that the devices would go through is more of a mucus type of solution, and we have an exact test soil that mimics a mucus solution. And there are times that it may nick a part of the eye that could produce a little bit of blood, and so we usually add about a 10% of a blood solution to that. And so we do mixtures of mucus and blood solutions to get exactly the soil that would be utilized in the surgical procedure, so that we make sure that we're exactly mimicking the surgical procedure.
That makes sense. There's a few different questions here that are coming in that are kind of similar. One about multiple groove designs, one about multiple spring types. How does either the groove configuration or the spring type impact the ability to clean the spring, via the procedure?
Well, our first baseline test showed no impact when we used 3 different commonly used groove configurations, which is a flat bottom or a V bottom or a taper bottom grooves. So as far as we know right now, we've seen no negative effects of those different groove configurations in a 5mm bore, of course. So far so good. We're going to continue our testing to find out more about that.
Yeah, the samples tested seem to show springs in an unconstrained or a non-deflected state. So does the spring have to be in an unconstrained or non-compressed state in order for the cleaning to be effective?
Well, for this test that we did it in an unconstrained state, where typically they'll disassemble the devices and then put them through the cleaning process. So I would say I would probably prefer to have it in an unconstrained state where there's no shaft and housing assembled. You disassemble so you have good access of fluids coming in through the groove. So I would definitely say unconstrained would be your best bet.
Okay, great. Let's do just a couple more questions here. Generally speaking, how does changing the spring coil height, coil width, and the ID impact spring cleanability, Steve?
Well, we've only tested one size of spring and plan on to expand into other sizes in the future as we develop our baseline testing matrix for canonical springs. I can't answer that right now, other than it's just the test that we've done originally. So hopefully later on we can find out more about how our cross-section sizes and wire diameters and spacings can make a difference in cleanability. Right now we don't know.
Okay. And then let's finish with one for Emily. Emily, how do you determine what contamination method is appropriate for the cleaning validation?
Yes, so the contamination method deals exactly with the surgical procedure, and so we want to make sure that we're mimicking that exactly. And so we, for example, if it's a heart procedure and about 2 inches of the device goes into the chest cavity, we want to soak the device, or soak the device in that much of the blood soil, and then we'll also take blood and put it on surgical gloves and handle the device as appropriate as the surgeon would handle the device on the handle, and also open and actuate the device appropriately.
Great. All right, well, if you have additional questions that come to mind, you're welcome to email those to me at pheaney@wtwhmedia.com. If we didn't get to your question, Steve or someone from Ball Steel will get back to you in the coming days with a personal answer, I promise. And thank you, everyone, for attending this webinar from Design World and Ball Steel Engineering. This presentation will be emailed to everyone in the coming days and will also eventually be available at www.designworldonline.com. Thanks once again to Steve and Emily for all of their insights. Have a great rest of your day, everyone, and goodbye.